When the human genome was completed, was I surprised that we're made only 23,000 genes? Absolutely. A worm that we go fishing with, 75,000 genes. A plant, 150,000 genes. So the question was, how we can explain the complexity of where we are.
We are here to celebrate the completion of the first survey of the entire human genome. Without a doubt, this is the most important, most wondrous map ever produced by humankind.
We were told by well-meaning people that we know the parts list for humanity. When you say the box is this, then enough people listen and they say, oh, the box is this. But the size of the box is just getting bigger and bigger.
How can we be so different biologically and physically from a mouse when our genes are 95% identical? It was clearly that the human genome was the tip of the iceberg.
We learned a lot over the last 50 years. We've cured a lot of problems, but there are these diseases of human progress, the ones that elude us, the Alzheimer's, the autisms. You have 2 or 3 people that look exactly alike from the same parents, and nobody disputes that there are genes for eye color and hair and how tall you are. But what we're more interested in is, well, why is one kid good in math and the other one not? That one kid can knock a golf ball 300 yards and the other one can't. There must be other elements that drive these circumstances.
There is this ecosystem that's a major and previously uncharacterized part of us that is playing this major role.
The analogy I've often thought about is how we went from the Ptolemaic to the Copernican view of the world. At the cosmic level, the macro level, it was really hard to say that, man is on this planet, just part of a much larger picture. Now we're saying man is affected by, maybe even ruled by, these microscopic we can't see. That's a really hard thing to incorporate.
What we're seeing today is a paradigm shift of our understanding of what's actually going on inside our bodies. And this is a completely new way of thinking about human health.
The human microbiome is the vast collection of microorganisms that inhabit every surface and cavity of the human body.
We harbor around 3 to 6 pounds of bacteria. And to put that in perspective, the human brain weighs 3 pounds.
There are many different life forms within the microbiome. It's viruses, it's bacteria, it's parasites, fungi.
Not only has the microbiome got some 9 million genes in it compared to our paltry 25,000, but it also is our interface with the world.
So what we're talking about here are literally trillions of microbial cells that are living on our skin, in our gut, in our nose, in our airways, in our urogenital tract. What we see as the outside world, outside our body, is through this microbial filter.
The first thing to say is that the mother has a microbiome, and the first big encounter with microbes is at birth. So in natural birth, since the mom breaks membranes during labor, the baby is exposed for all those hours.
When you watch a child born, the child is one with mother and is literally drinking mom's fluids. The skin is getting colonized. As the baby's breathing, the lungs are getting colonized by bacteria and microbes, viruses.
We interrupt this process. A baby was born by C-section, given formula instead of breast milk, exposed to antibiotics. We also raise the kids in extremely clean environments. We went to the Amazon and studied the microbiome of very remote people. And we were astonished to see they had almost twice the bacterial diversity in their guts in relation to the healthiest Americans.
Microbiome research suggests that over generations, we are losing organisms. diet and sanitation and other advances in lifestyle, all of these have been shown to impact the microbiome.
So the whole combination seems to be not good, and we suspect that is helping to drive the modern epidemics that didn't exist before and that are increasing as countries develop.
There have been so many studies published on the association between changes in the microbiome and disease. I would say that it's difficult for somebody like myself, who's a microbiome scientist, to even keep up. I guess we could just start with the idea of inflammation as a component of chronic disease.
There's a constant state of inflammation in our body, but when inflammation runs away with us, we can have many different conditions like eczema, allergies and asthma, also IBD and inflammatory processes in our gastrointestinal tract, Crohn's disease. These are problems of overactive inflammatory responses.
The capability to lose tolerance or stay healthy, it really depends how the immune system is trained during the first 1,000 days of life. And the trainer, of course, is the microbiome.
We have been studying a flavor of immune cells called a natural killer T cell, and it's very important in a wide variety of complex diseases. We looked at the germ-free pup that was born to a germ-free mom, and we saw that the germ-free pup had a massive increase in this NKT cell that's there to sense lipids, much greater numbers than the mice where the mom had bacteria in the birth canal. There's a developmental event that's going on that the microbes give the immune system the proper kickstart to evolve in the right direction.
If you have a balanced microbiome, so in other words, you're born the way you're supposed to, you are being fed food and other junk stuff that we give to our kids nowadays, you non-abuse of antibiotics. So in other words, you keep the microbiome in balance. The microbiome will teach the immune system to unleash inflammation only when we are really, really under danger. So the threshold is put up there. But if you are out of luck and you have, everything wrong, you will have an imbalanced microbiome, so what we call dysbiosis. This will put the threshold for the immune system to unleash inflammation, even for trivial reasons. That increases the chance in doing that sooner or later this will go out of control and you will develop these problems.
One of the most interesting pieces of this field is this history of these, as they're called, fecal transplants. And as disgusting as they sound, they are exactly as they sound, of taking feces from a healthy individual and putting them into the colon or large intestine of someone who, say, has a disease. And what's fascinating here is if you take someone with Clostridium difficile infection and you take feces from someone who doesn't have it, you give it to the person who does have it, you cure them, clinically cure them, in most reports over 90% of the time. And that's phenomenal. Most antibiotics don't seem to work at that level. And that kind of demonstration, while it's most robust in Clostridium difficile infection, is being done in several other things. So there's people who have been doing a series of fecal transplants for people with ulcerative colitis in particular. There's people looking at irritable bowel syndrome. We're now understanding that you can transmit food allergies. It turns out you can transmit obesity. And who knows what else.
In our studies on obesity, what we found was if you get a fatter mouse from the mouse that gets a fecal transplant from the fat twin, a thinner mouse with a fecal transplant from the thin twin. So you can induce obesity with diet and then transplant the microbes from that altered ecosystem, transplant those into a new mouse and make that new mouse obese. But what happens is that the altered microbial community actually makes the mouse eat too much, so it's hungrier, that's why it becomes obese. You can cure it with antibiotics. You can cure it by putting in its cage what a normal mouse would eat, so it's hungry all the time, but at least it's not fat. And you can also transmit that microbial-induced hunger to a new mouse that's genetically normal by transmitting the microbiome. 12 years ago, we found the first associations between obesity and the microbiome, and what's been amazing is how rapidly that's extended to other diseases.
What is most surprising to me is how in some cases, single organisms, presence or absence, can define what seem to be otherwise very complicated states. And I'd say perhaps the most tangible example, which we all take for granted, is a particular bacteria found in the stomach of some people, Helicobacter pylori.
We and others showed that Helicobacter was an important cause of stomach cancer. Now, at the same time, we looked at esophageal reflux, that is now the leading cause of esophageal cancer. And I thought maybe we'd find an association with Helicobacter, but we found the opposite. We found an inverse association, suggesting the idea that Helicobacter might be protecting against reflux. So a more complex story is emerging. Helicobacter is bad for some people, seems to be good for other people.
I just find that fascinating because cancer is such a complicated disease, so that single bacteria defining that state is really powerful in my mind.
There was one bacteria that seemed to be enriched in the colon tumors versus people's normal, healthy colon. And it's a bacterium, Fusobacterium nucleatum, that we have in all our mouths. We found that it was living in the tumor, in between cells, and within cells. Maybe somewhere between 10 to 30% of colorectal cancer patients seem to harbor this bacteria, and a lot of it. And actually having Fusobacterium in your tumor was worse for you in terms of your prognosis. We focused on the immune cells in the tumor, and we found that Fusobacterium nucleatum make a protein that binds another protein on immune cells. And those immune cells are natural killer cells and other tumor-infiltrating lymphocytes. And when that molecule, called FAP2, binds a molecule called TIGIT on a tumor-infiltrating lymphocyte, something really interesting happens. It sort of puts the brakes on that immune cell from killing.
De ce atât de tineri? De ce în culmea succesului? Cum a fost posibil să moară când aveau totul? Întrebări ce rămân fără răspuns. Prea tânăr pentru a muri. De vineri până duminică, la 22 pe IGIENA.
We have a lot of other ongoing projects in the lab thinking about and focused on all the different ways in which Fuso is affecting the immune system. And if we know those things, if we know the molecules that it uses to interact with the immune system, if we know the molecules that it needs to live in a tumor, those are all targets. So we are engaging in a lot of screens to find genes and proteins and other molecules made by the microbiome that make response to immunotherapy better.
What's interesting is if you look at the microbiomes of many people, there's a lot of differences in the specific organisms that make up the microbiomes. If you look, however, at the same microbiomes from a functional perspective, it seems that the microbiomes are actually doing something consistent across all of these people. And so from another perspective, we like to think about the microbiome as an organ, and that gives you a basis of kind of thinking about points of action, how to engage. Now, the microbiome is part of an organ system with the immune system, and you put that together and you find these two complex pieces of the human body that basically shape each other, train each other, inform each other, influence each other. You now can see the microbiome as a really powerful way to engage the immune system and everything that that's involved in, whether it's autoimmune disease like multiple sclerosis, food allergies, or cancer.
Fatigue is a very difficult word in this illness because it's not the fatigue that you and I would feel because we went on a long bike ride. It's that feeling that you're sort of walking through glue, that you just have no energy. Many of these patients have to be in darkened rooms and actually wear ear protection headphones to mitigate the effects of the sound. Those people are the most severe, and they are invisible to the public. Most people who get chronic fatigue syndrome report that they had a flu-like illness from which they never recovered. So if you can imagine when you have the headaches, the muscle aches, the general malaise, the fatigue that comes with the flu, that's what people with this illness have, except that they can have it for 40 years. They have all the symptoms as if they are being attacked by a pathogen, and yet there's no pathogen that can be found. What we found is healthy controls had more species present in their guts than people with chronic fatigue syndrome. And many of these species were known to have these anti-inflammatory effects. And if your intestinal lining is not properly functioning, some of the bacteria can actually get out of your gut into your bloodstream and cause your body to think that there's bacteria in my bloodstream, so I better mount an immune response to this. Now, I'm often asked whether people can take some probiotics to compensate for this problem. And one possibility would be for some studies to be done in the future to see if there are particular species that could be supplied that would help.
How does introduction of a probiotic into this gut ecosystem change the function of the organisms that are naturally living there? We can begin to get some good insights just by looking at changes in relative abundance of genes. What we have shown is that a specific lactobacillus probiotic increases the gene expression profile in some of the key microbes in the GI tract that produce butyrate. And butyrate has been described as a very potent anti-inflammatory molecule. These butyrate producers are only able to produce butyrate if they're fermenting some of the complex polysaccharides that you ingest in fruits and vegetables and whole grains. So if you're eating sugar-coated cereal, you're not even giving these butyrate producers the starting material that they need to produce butyrate. So you could be taking a probiotic, but if they're not able to produce butyrate, then what's the point? So the ability of the microbiome to influence inflammation in the body is critically dependent upon what we feed our microbes.
It turns out that certain products that bacteria make, like short-chain fatty acids and other, take your host cells and they can affect their DNA methylation. So what I eat can affect what my bacteria make, and what my bacteria make can affect what genes are turned on and off in my body. Weird.
The nerves in our body, this network of electrical connectivity that goes from our brain, a massive clump of neurons, all the way down our spine and then spreads to every part of our body. And microbes affect that too. They produce neurotransmitters. They consume neurotransmitters in the gut, for example, and that can affect the signaling pathways throughout this network of connectivity in our body.
The majority of the neurons outside of our brains are actually in our gut, about 70% of them. And there's literature that goes back 15 years that suggests that perhaps neurons are dying in the gut, in the intestine, years before they're actually dying in the brain. And so this suggests to us that perhaps bacteria in the gut were playing a role in the neurodegenerative process. What we've recently shown is that if we take mice that don't have gut bacteria, but the mice genetically are predisposed to developing Parkinson's, in the absence of gut bacteria, they don't display motor symptoms. They also don't have the underlying pathology or the brain changes that are shown in Parkinson's. This result tells us that there are organisms that are driving these symptoms. There are a subset or maybe even a handful of organisms that are promoting pathology, promoting disease. We strongly believe there are other organisms which are protective. And I think what microbiome research has told us is that the balance between, let's call them good and bad bacteria, is integral to the disease process. There are now about 24 studies that show that the microbiome of children with autism are quite different than the microbiome of neurotypical children. There are different organisms, and those organisms that are shared are there in different ratios. And within those organisms we tested, we identified one organism that appeared to have an effect on behaviors associated with autism. It would be incredible if we can just give a pill to an individual that has a live bacteria and somehow improve, maybe even eliminate their autism or other behavioral deficits. If there's ever a treatment, and I believe there will be a treatment for autism someday, it will not be universal. You will have to personalize treatments for different individuals.
This, again, is the promise of personalized medicine, that I will be able to extract material from your body, analyze it in a laboratory, and tell you how well you're going to play golf, whether you're going to get into MIT or not, et cetera, et cetera. And I think it's a long shot at this point because there's just too many input variables in our measurements. We need the movie, not the still picture.
Some of the things I've actually been surprised by in terms of personalized microbial diversity as we've worked in the area is the degree to which, to a pretty good approximation, everybody has a different strain of every microbe. So not only do two individuals, even from similar populations, tend not to share the same microbes, even when they do, the specific strains, the individual genetic variations within a particular microbe tend to be personalized to you as a host. So we've looked, for example, at adult twins, and they do tend to share more of the same types of microbes than unrelated individuals. But even in those twins, those microbes seem to have diverged over time, so that by the time you grow up, you have acquired different microbial genetic elements and DNA and functional potential than your twin. Of course, this gets even more and more different between different individuals and different populations, to the point where even saying how many species are in the human microbiome is very difficult. Are we talking about the microbiome of people in this city or in this country or in the world?
Most of the diversity in the system are actually the phage, which are the viruses that infect the bacteria. You get these phage when you're young, and then they evolve in just this one particular protein to stick to your mucus specifically. So what you've got in your gut are all these phage that are explicitly holding onto your mucus, but not onto mine. What it's going to do is it's going to attach to a bacteria, inject DNA into it, and take over. Usually the bacteria dies. So it's a phage-based immune system. This is extremely important because you can imagine we can just use phage to kill bacteria. And what's nice is the tail of the phage is specific for that bacteria type. So you don't have to kill all the bacteria, you can just kill the one you're looking to kill. There was a doctor over at UCSD who just did a successful phage therapy treatment against one of the most antibiotic-resistant bugs we know of. He got a whole bunch of phage from different people, and it totally worked. Remember, the phage are binding to the cell, the outside the cell, which means that if you're going to protect yourself against the phage, so if you're a bacteria, you have to change your cell somehow. Whatever the bacteria that survived the phage did to protect themselves made them sensitive to antibiotics again. Isn't that cool?
If you take even a well-studied microbial community like the human gut, generously, the fraction of gene products that we understand are between, say, 25 to 50%. So there's a pool, even in the gut microbiome, of microbial dark matter. I've been able to use the Donald Rumsfeld quote for several years now regarding known unknowns and unknown unknowns. And I think some real progress in the microbiome has ironically been in moving unknown unknowns to known unknowns.
When you're faced with something that you really hadn't defined, then you have to back up and say, wow, all bets are off here. We kind of have to look at the system in a fresh light.
When you talk about complexity, you have to bring to a reductive model of human genome and say how we can be so complex with only 23,000 genes. Well, let's say that these 23,000 genes are notes on a piano. So you have 23,000 notes. And now you have a piano player. That is the microbiome. now the combination will become infinite. Infinite.
Richard Feynman, the famous physicist, he said, whatever is not surrounded by uncertainty cannot represent truth. Whatever is not surrounded by uncertainty, a lot of negatives in there, it can't be true because everything's uncertain. And here we are. That'll be a good ender.